organelles in them because they don't exist, and so they typically look very homogeneous or maybe somewhat granular, and that's because they lack these membrane-bound organelles, and they and sometimes you can also see some, or typically lack these empty-looking spaces. Also, typically with many of the cyanobacteria, they're surrounded by a mucilaginous sheath. This mucilaginous sheath can extend sometimes quite extensively from the individual cells.
And we'll see some photos of those in just a little bit. Okay, now if you reach back into Principles 1 and 2 and probably other biology classes that you've had prior to coming to Carroll, you've probably focused on the Monera, or the prokaryotic things, and you probably looked at some examples under a microscope, and you probably learned about 3 different cell types or cell shapes. We have coccoid, which refers to the ones that are kind of circular like this. We have sort of longer sausagey looking things, which are the bacilla. Is it bacilla or bacilli?
Bacilli for plural.
Bacilli, for plural. And then sort of these long corkscrew kind of squiggly ones, which are spirilla. Oops, misspelling. Spirilli, right? But we're going to be looking at, and typically most monerans that you look at will be very, very small. The thing about the cyanobacteria that we're going to look at, a general characteristic of this group, is the individual cells are truly ginormous. They're gigantic on the scale of most other prokaryotes. And so they're relatively easy to see. For the most part, we're not going to need to go to oil immersion for any of the things that we're going to be looking at in class. We can see them easily at the lower magnifications with our microscopes. But look at the different cell types that we have in these examples. Here is a cell type on the far left. These cells are kind of coccoid, kind of bacilli. They're sort of intermediate. We can look at cells here. This is an example of a genus we'll be looking at today. You can see these sort of squashed, kind of flattened cell types. Doesn't really clearly look like coccoid, bacilli, or spirilli. And then here we have an example of something else we'll look at today, where we can see the individual cells. And I'm going to pick a different color here on my pointer so that we can see these a little bit better, hopefully. Right here is one cell, this short, fat, squatty thing. We have its neighboring cells. They're each very different from the typical coccoid, bacilli, or spirilla cell. And what we really want you guys to focus on in this class, when we make our observations in class, is really look at and understand the basic characteristics of organisms. So this is all about developing your observational skills. And some things that we can note about these, even though they're really simple cells, is the relative length of the cells versus the width. These cells are very much wider than they are long. And these are kinds of characteristics that phycologists, people who study algae, will pay attention to, is the anatomy or the morphology of individual cells. Yeah.
So here we have Arthrospira. This is the genus Arthrospira, and this is a really great example of a cyanobacterium that looks really granular. A lot of the specimens that we showed you on the previous slide were much more homogeneous in the way that they appeared, but Arthrospira looks much more granular in appearance, and this is because it has all of these gas vesicles that help to keep the trichomes, and the trichomes is this whole, all these cells that are linked together into a trichome, and those gas vesicles help to keep the cyanobacterium afloat in the water column.
So we're going to ask you guys to pay attention to and illustrate the anatomy of the cells, the shapes of these, how they're sort of arranged relative to each other, but also to take careful observations of the contents of the cells. So in this case, we have a trichome, which is that line of cells. The cells are wider than they are long, and when you make your illustrations, you want to talk about how there are little granules that you can see in open spaces in the cells.
So here we have different examples of coccoid taxa. Some people also say coxoid taxa, so kind of a tomato thing. These are all examples of organisms within the Crococcales. And one of the characteristics that I really want to point out is we can see it really well with Gleocapsa, which is the organism that you see in the center. So you can see the individual cells, and then all of the cells are encapsulated in mucilage.
So sorry to interrupt. These little things I'm tracing right now, these are the actual cells, and then this is each cell surrounded by a layer of mucilage.
And then you can also see an even larger area where you can see then they're encapsulated in a glob of mucilage, and then this whole set of cells you can see is also encapsulated in mucilage. So Gleocapsa is a really great example of a cyanobacterium that has this really thick, multi-layered mucilaginous sheath.
With many layers.
Mm-hmm.
Okay. I'm spacing out on what we're going to talk about here.
So then these are filamentous taxa.
Right, so here we have filamentous taxa. A filament, so we talked about a trichome, a string of cells, right, of cyanobacterial cells is called a trichome. So in this example of Oscillatoria, we can see cells that are wider than they are long. They're all packed in together here. That is the trichome. The filament would be the trichome plus the mucilaginous sheath. And so here we can see leptolingbia, and in this case, the magnification isn't equivalent between these, but we have cells that are almost as long as they are wide. And Oscillatoria is one of the species that we'll be examining today. Do you want to talk about Nostocales?
Sure. So another order that we're going to be looking at today is the order Nostocales. And these organisms, these cyanobacteria, are different from organisms in the Oscillatoriales because they have some very unique cell types that are only associated with this order. And here we have 3 different examples of genera that belong to this order. So Nostoc, which is on the far left, we're going to be looking at either Nostoc or Anabina in class. So Anabina is in the center, and then Cylindrospermum is on the far right of your screen.
Do you want to start with the vegetative cells?
I'll start with the vegetative cells. So the vegetative cells are what Dr. Scheel is outlining right now. And these vegetative cells, their basic job is photosynthesis, so oxygenic photosynthesis. Another cell type that we find only within the order Nostocales are the heterocytes.
Labeled with H, and I'm going to trace these out. So, oops, here is a heterocyte.
Now, if you look very carefully at the heterocytes, you'll notice some difference between the heterocytes and the vegetative cells. First off, the vegetative cells in most of these examples look a little bit more granular in appearance, whereas the heterocytes look very, very clear. A second distinguishing and really important characteristic are the presence of what are called polar nodules. They look like little commas, and typically they're found on either side of the heterocyte. The job of the heterocyte is really important. The heterocyte functions in nitrogen fixation. So you might ask, well, what's nitrogen fixation? Nitrogen fixation is the process by which atmospheric nitrogen is made into an inorganic form that can then be assimilated into organic compounds. And what's really important about this process is that only specific prokaryotic organisms have the ability to fix nitrogen. So no eukaryotic organisms that we know of have the ability to fix nitrogen. So for example, the nitrogen that is in the proteins in your body and nucleic acids and any sort of nitrogen-containing compound in your body, at some point that nitrogen was most likely fixed by some prokaryotic organism that made its way through the food web.
Yeah, so tying this to chemistry, something that you may be familiar with already is that molecular nitrogen is 2 nitrogen atoms that are strongly bound together. Only certain groups of organisms have the ability to break those chemical bonds or those bonds between the 2 nitrogen atoms, and then, as Dr. Janowski was saying, incorporate that ultimately into organic material. So that's a big deal.
Yeah. The other cell type that we want to point out is what's called an akinete, right? And that's what Dr. Scheel is labeling right now.
This big fuzzy thing right here.
Yeah, typically akinetes
Here's another one.
Will look a little bit more granular than the heterocytes. These akinete are a resting cell. So, for example, a lot of these organisms are aquatic. So imagine that you have some pond or even just some puddle that starts to dry out. These organisms, because they're aquatic, the vegetative cells require an aqueous medium to survive. And so if their environment starts to dry up, they can form these resting cells known as akinetes. And the akinetes are very, very resistant to any sort of environmental change. And so even if that pond or that puddle or whatever it is completely dries up, that resting stage of that akinete can survive. And then when the conditions change, that akinete can what's called germinate. Of course, it's not germinating like a seed, but it's akin to that in that it's leading to the development of a new organism.
Okay. In the actual lab that we have, we're